Hi everyone, and thanks to the organizers for giving me the opportunity to present my poster entitled Mutation-Related Apparent Myelin No Axon Density Drives White Matter Pathology in Pre-Manifest Huntington's Disease. This was part of the work I carried out at Cardiff University for my PhD and was built on a body of evidence showing subtle and progressive white matter alterations early in HD progression. I was particularly interested in understanding a little bit more about the etiology of white matter changes in HD. And specifically, we know that white matter is made up of both myelin and axons. And so we don't know whether myelin, axons, or both are impaired in HD. And I was also interested in understanding a little bit more about the role of white matter changes in HD pathogenesis and progression. Why should we care? Well, we know that HD cannot currently be cured, and assessing early white matter microstructural changes might prove useful for identifying disease-related biomarkers, and so give us power to improve prediction and diagnosis, and also improve measurement of response to therapeutic approaches. What we did in this study was to exploit the ultra-strong gradients of the connectome scanner, which allowed us to better differentially attenuate the MRI signal coming from the intraaxonal space versus the one coming from the extraaxonal space. And specifically, we assessed white matter microstructure in HD by combining standard DTI measures with an MRI proxy measure of axon density, or FR, from the Charm model of diffusion, and with an MRI proxy measure of myelin, or MTR, from the magnetization transfer imaging. And this measure has been shown to present a high correlation with histological measures of myelin, as shown here in this review by Mancini et al., published last year. We assessed 25 HD carriers and 25 age and sex match controls, and the sample characteristics are presented in this table. And we use the diffusion data to perform tractography and thus to virtually reconstruct white matter pathways in the brain, and then to further isolate the corpus callosum and segment it into 7 different portions. So as the callosum presents smaller late myelinating fibers at the front, while later larger earlier myelinating fibers are present at the back. Here I was specifically interested in understanding whether there is a topological specificity of changes associated with HD. We then use tractometry to project all of these different microstructural parameters across the callosum. And hopefully you'll see here how each microstructural parameter presents a different spatial distribution and should therefore give us complementary information. We use PCA to reduce data dimensionality and extract 2 components, an axon density component and an apparent myelin component. And we then assess the effects of group and segment on these components. We then also used PCA to extract a composite cognitive component after assessing HD patients on a series of cognitive and motor tests. And this is because we were interested in understanding a little bit more about disease-related brain function relationships. And so we use Perman correlations to assess relationships between our white matter components, cognitive scores, as well as CHG repeat size and DPS. What we found was alterations in callosal apparent myelin of HD patients before changes in axon density could be detected. And specifically, as you can see here, there was an overall higher apparent myelin in the more anterior portions of the callosum, while apparent myelin was lower than controls in more posterior portions. Importantly, we detected a positive significant association between apparent myelin in the callosum and CHG repeat size, but we didn't detect a significant association with disease burden score, and the association with the cognitive performance did not survive multiple comparison correction. We then corroborated these findings with tract-based cluster analysis, which It's a novel whole brain assessment which combines anatomical and cognitivity information provided by tractography with the spatial encoding of voxels. And again, consistent with the PCA analysis, we detected a decrease in apparent myelin in the posterior portion of the callosum. And additionally, we also detected an increase in the proxy measure of axon density in HD patients compared to controls in the left cortical spinal tract. To summarize, we detected significant alterations in callosal apparent myelin of HD patients before we could detect any changes in apparent axon density, suggesting that white matter changes are unlikely to be just a consequence of Wallerian degeneration. Additionally, these changes in apparent myelin were shown to present a topological specificity, so that apparent myelin was higher than healthy controls in more anterior portions and lower than healthy controls in more posterior portions of the callosum. We also detected a significant association with CAG repeat size, suggesting that myelin alterations are probably a direct result of the mutation rather than just being a consequence of disease progression. And thus we suggest that it is possible that the HD mutation leads to excessive myelin production earlier in disease progression, and that these increases later on lead to detrimental effects because of oxidative stress. Outside the callosum, we detected other changes which likely reflect axonal alterations, such as axonal swelling or reactive gliosis. In this last slide, I'd like to thank all of the people that made my PhD possible and this work possible. Thank you all for listening, and I'm happy to answer any questions. Thank you.